It's my great pleasure to introduce today Dr. Bror Jönsson. And his academic journey was quite interesting. He's moved through several fields to where he is today doing remote sensing and ecosystem modeling. He started with his bachelor and master's at Uppsala University biology. He then moved to Stockholm University for a second master's in coastal biogeochemistry, and then stayed at Stockholm for his PhD in physical oceanography. So he's covered a number of bases within oceanography. Then he left Sweden for a postdoctoral appointment at Boston University here in the States, followed by another postdoctoral associateship at Princeton. He remained at Princeton for a number of years. And then he went back to Europe, to the UK, at Plymouth Marine Lab for five years. Yeah. And then he came back to us in the States. He's currently a research assistant professor at University of New Hampshire. Without further ado, Bror, welcome.
Great, thank you so much. I hope you can hear me. I'm going to go through a lot of slides, but I'm going to try to go through a couple of concepts. If anything sounds weird or you have any questions, you can always stop me and we can start discussing wherever you want. I have an idea where we want to go, but we don't have to end up there after this hour. So what I'm going to talk about now is biological production in the ocean. First of all, I just want to see, just raise a hand, how many people here would use the word physics when they describe themselves in any way, form or shape? Like physical oceanographer, meteorologist? Couple, good. How many would use in some way the word biologist when they talk to their parents? All right, most people are biologists. Chemists, any chemists here? Couple. Even geologists? Nah. Or anything else? Okay, good. Then just to know how to put focus on what we're talking about. Great. So the basic idea here that we're interested in is basically carbon fluxes in the ocean. That is what we are, as a research community, very, very focused on. And this is because of climate change, it's because of how we are using coastal resources in the ocean, how to understand fisheries, etc., etc. Almost all of biological oceanography, or a lot of biological oceanography, when we are explaining to people why they should give us money to work on it, we end up talking about carbon fluxes in the ocean. And very simplistic, it looks like this. We have what's called gross primary production, which means that phytoplankton, green bugs in the ocean, are taking up CO2 and turning it into organic carbon. And then NPP is net primary production, which means that they're taking some of that carbon they've created, because this is now a proxy for energy, and they're using it to live. The rest is being used to grow, and that growth is what we call NPP. And then everyone else wants to eat them when we get net community production, which is how much the community, as in phytoplankton, viruses, whales, are growing, expanding. Nothing can expand forever, so at some point we will find an equilibrium and things will start to go away or being exported out of this ecosystem, and that's where we get what's called export production. And this is a very key parameter that we want to get to. It's very hard to measure, and I'm not going to talk so much about it today. Instead, I'm going to talk about these two components that we are very interested in. They're very hard to distinguish between each other for different reasons. But basically, it is how much carbon phytoplankton take up, and then how much they are able to use that to grow. And the reason why it's hard to work with is because it all comes down to the timescales of definition. Are we talking about hours? Are we talking about years? Are we talking about a liter of water? Are we talking about the entire ocean? The other thing I'm going to talk about and focus on now is how to estimate these parameters using satellite. So satellite-derived primary production is kind of the focus of the talk. I'm just going to do a little side here and talk about remote sensing and satellite data. And what I'm focusing on here primarily is what's called ocean color. Ocean color means that we're using ocean, basically we're looking at the color of the ocean. And we have a number of satellites that are flying that can give us this information. So think about a camera, but instead of having 3 colors, we get tons of different colors. These satellites have provided us with an amazing ability to understand the ocean in a way that we couldn't do before. And this is an example. This is sea surface temperature, but we have the same thing with ocean color. This is the amount of data we get from a month with all the assets we have for sea surface temperature. This is how much data we get from a satellite in a day. So suddenly we are actually able to get a really broad understanding of how the ocean works, at least the surface ocean works. And the way it works is, as I said, is that the sun is shining onto the ocean, and some of this light is penetrating into depth. In the depth, at deeper waters, we have things like phytoplankton. They're reflecting back this light, and the satellite can see the light that's been reflected up. Different colors are reflecting differently, and therefore and there are tons of different processes that are very complicated for this. But basically, the differences in different wavelengths being reflected will create different lights. And we can see this in this picture. For example, in this case, if we go from the very near shore out to the open ocean, you can see that the water is it's literally changing color. And this is the spectra of the light. So you can see that the spectra is also changing because of that. And using this information, we can figure out different properties of the ocean. The most important one is chlorophyll. So everything that is green and is living has chloroplasts that are helping in assimilating light into carbon. And we're using that, literally the color of these organisms as a way of estimating how many they are, so we can get biomass. And from that biomass of chlorophyll, we can calculate carbon content as well. I shouldn't say we do, but there are a lot of issues with it. And with these systems, we can create these very nice images of where we have a lot of phytoplankton, where we have a lot of productivity in the ocean. Because to create trends was very useful. This just shows how that some areas over the last 25 years have increased in chlorophyll and other areas will decrease. The other parameter that's very important is called attenuation. But that means how far the light can penetrate into the ocean. This just shows an example of that. Two of the very important features here is like where we see about 10% of the light from the surface. That is what's called the first optical depth. This is how far down the satellites can see. Anything below this, we don't know anything about from satellites. The other level, which we as biological oceanographers love, and no one really knows why, is what we call the euphotic depth. This is the level of 1% light, because we've decided that this is how far down organisms can grow, which is probably not true, because there's some differences in how much light there is, but we're using this as an estimate of like the productive zone of the ocean. With these features, with these different satellite parameters, such as light, as we can get temperature, we can calculate the growth rate of the phytoplankton. And then if we have the biomass in one way or another, we can try to calculate the primary production. This is the simplest way to do this. There are much more complicated models where we have very elaborate light models, and we have to figure out how to deal with the depth and so on. Complicated. But all these models, all these systems, and this is something that has been focused on in biological oceanography for the last 50, 60 years. They are all assuming steady state, like nothing is really changing on the short timescales. That all the production that is happening is also consumed, so nothing is increasing or decreasing in whatever system we're looking at. And this works if we're looking at long time series, monthly to seasonal to climatologies, etc. Great, everyone's happy. We can now estimate primary production. We can see how the world works. Everything is great. We can create these very nice images of climatological primary production in this region. So we don't have to do anything more. It's solved. Great. Nope. Because scientists are never happy, and we are always finding other things to work on. And that's where we're coming in. And the reason why this is complicated, much more complicated as we have been looking at before, is that different processes are working on different time scales and different spatial scales. So there are many of these images, but this just shows that different physical and biological processes are working on different spatial and temporal scales. And normally we are focusing on these climate change, seasonal scales, but there's an area here where there are a lot of very important processes that we also want to take to understand. And those assumptions that we did doesn't work on those timescales. And one of the reasons why these scales are becoming very important is that we want to have higher and higher resolution on the ocean models. This is an example of this. This is a temperature from a model from DFDL in New Jersey, and this is a satellite image. This is chlorophyll from the same models, and this is the satellite image. And you can see that the large scale structures look really, really similar. I was impressed when I created this image. I kind of created this to show my postdoc advisor how bad his model was, and I had to eat crow from that. But you can see that in these regions near the coast of California, there's a big difference in the model. So there are certain processes that these large-scale coarse models are not picking up. So therefore, we want to have the models to have higher and higher resolution. If we have higher resolution in the horizontal, we need higher resolution in the vertical. This is just a kind of a toy model we ran, where the white boxes we can see here are the same resolution as the model I just showed you. The kind of smoother field is based on a model that has a 1/4 degree resolution. These white boxes are 1 degree resolution. And then we take a very high resolution 1 kilometer degree model here that we have laid in. And you have the horizontal at the top, and then you have the vertical. And what you can see is that we get much more complicated horizontal processes resolved. But we also get much more complicated vertical processes resolved. So when the horizontal resolution increases, the vertical dynamics get much more complicated. And what happens then too is that we have to increase the temporal resolution. Because any model where it has a domain that they work well. If you have too high resolution, when I say model now, I mean a GCM, so global circulation model, general circulation model. Just as ocean models, you have a modeling group here that sometimes give presentations, and you've probably seen those. But anyway, so if you have very high resolution, you get numerical noise. You will get output from the models that doesn't make any sense. Then you have a favorite domain where you have really good scale, and then if it's too long temporal resolution, for temporal scales, it doesn't work again. Another problem why we need to think about how we do these satellite productivity estimate differently is because of temporal lags. So what I did here was that I took a very simple biogeochemical model of a growth model of phytoplankton, where we start with a lot of nutrients and no phytoplankton, and then the phytoplankton starts to grow, and they eat up all the nutrients, and then the zooplankton starts to grow, and they eat up all the phytoplankton. This is like very traditional, very simple, very kind of a fun exercise to do. If you estimate primary production as we would do, like basically how fast do the phytoplankton grow, we get the blue line here. This is how the primary production actually looks like in this simple toy model. If we use these satellite productivity models that I just showed you and estimated primary production, we get the red line. So you can see that it's a very different description of how this system have changed over these days. The sum is almost the same. So we're not getting the wrong number, we're just getting the lag wrong. And then finally, one thing that is very important is that when we're thinking about biological system, we always have a spatial decorrelation. And think of a SoMAS, like the George's Bank or something, where you have productivity because it gets shallow. You get a very intense growth cell up here, and that would create this huge plume of biomass. If we use this biomass to estimate this growth, we get a decorrelation in space that is important. Finally, The region where these processes and these questions, these challenges are the most important and most hardest to work with are in upwelling regions. Most of you have probably heard about it if not in undergrad, but basically, especially on the western coast of all the continents, we have regions where we have certain wind conditions that are pushing up deep waters, nutrient-rich waters from the deep up to the surface, and we get very, very productive areas. These regions are some of the most important for fisheries and et cetera, in the world, and there's an intense interest in studying them. So we have worked on developing a couple of different ways of looking at primary production from satellite that I'm going to present. The first one is to use a Lagrangian frame. The idea here is to basically follow a water mass and see how it changes by time, rather than just sitting at one point in space and look at how water mass are moving by you. Think of the difference of having a moored station, like a mooring somewhere, and having a mooring you cut loose and see how it moves, that's moving around. And the idea here is pretty simple. We just take Lagrangian particle tracks. So we take particles and we see it in a physical ocean model where we have velocity fields from, and we follow them for a little time. And wherever they are matching up with the satellite image, we do that matchups. So let's say that you're in the model, you follow this particle, and day 1, you have a matchup with the satellite data. Day 2, day 3, nothing. Then day 4 and day 5, we have matchups. We just match them up. Now we can see how chlorophyll is changing along this path. And doing that for one particle is statistically ridiculous because there's so many uncertainties. But we're doing this in a computer. We can do as many particles as well. We can do hundreds of thousands of particles. What this means is that we now can follow these productivity across along these water masses, and we can see how they evolve. And we can use that. Then we can start to calculate how primary production is changing, how chlorophyll is changing, and we can use that as a proxy for primary production. We did this in the Southern California bight. We used specifically developed satellite fields from SCRIPS. We used the velocity fields from NASA, and we used the particle tracking code that I have been co-developing during my PhD. And this is the result. So what we see here is increase and decrease in chlorophyll. When you see red blobs, that means that we have growth of phytoplankton. When we see blue, that means that phytoplankton is decreasing, chlorophyll is decreasing. There are a number of reasons why it can decrease. But you can see that it's very episodic, it's very event-driven, and you can see that the distribution and it's incredibly local. We can take all these data from a full year and create an average. And that looks like this. On the left, we have chlorophyll, and on the right, we have sea surface temperature. So what this means is that when we have these green and blue cells, every time a particle passes through here, the temperature is decreasing. And then we interpret that is that this is where we have an upwelling cell. We can see in the chlorophyll that in the corresponding region, the chlorophyll is increasing. So what we're saying with this is that we can pick up where we have upwelling, and we can also pick up the corresponding growth from that upwelling. And this is really useful because we can now start to really see in detail where these processes are happening. One cool thing we can do with this is also doing extrapolation. Basically, if we have these particles that are being affected, and we have a model that doing forecasting, basically a weather forecasting in the ocean, we can just do a simple linear regression forward, and we can calculate what the chlorophyll will be in the future. This is something we've done around the United Kingdom. So this just shows all these like the particle tracks that we used. And then we showed the true sentinel, so true chlorophyll 3 days ahead, and our predicted chlorophyll 3 days ahead. We get an R-square of 0.81. So what we can use this for is, for example, predicting harmful algal bloom or predicting pollution events with water quality issues. So this is a really cool thing. Second, one thing we've been working on is trying to estimate primary production with these satellite proxies but instead of using the traditional models, using machine learning. In this case, a very well-established method called random forest regression. And this is a project we've done within this is work we've done within a project called the PRIMUS, or Primary Productivity and Upload System. If anyone is interested in biology and primary production, we have tons of data in this project. It was a pretty big ESA project we were running for a couple of years. So this is a nice thing if you're interested in collaborating. And basically, one of the things we have are tons of observations of primary productivity in Galicia, and in the region around the Canary Island upland. And I should say, basically what these observations are is that you do an incubation for a couple of hours. So you take a water and you put in radioactive carbon, and you see how that radioactive carbon is incorporated into the phytoplankton. And that way you can see how much carbon they're taking up. And you're also doing that at different depths. So you get a depth resolved profile of primary production. This method is barely used in the US anymore because no one wants to work with radioactive carbon. It's much more common in Europe, and this is a challenge. If we do this with the classical primary production models, these are like state-of-the-art primary production models that we're working on. And we look at 24-hour incubations. We don't get any correlation at all. It looks really, really bad, which is a problem. So this means that there's something we need to work. If we instead use 2-hour incubations, and we're working on, and we do compensate for day length and so on, so we get a better fit, but it's still a bad fit. And I told you that when I showed you satellite-derived primary production, I showed you primary production versus GPP versus NPP. What this suggests is that these primary production models that are sold as NPP are more doing GPP. So they're picking up how much the primary production, how much the phytoplankton is assimilating over the day, not how much they are growing. But it's the growing part we want. It's not necessarily assimilating. So that's an issue that we need to think about. But this really shows that the question about what we are measuring with these primary production models is something to sit down and think a little bit more about. So can we do this with random forest regression as well? Well, yes, we can. So we can start with just using chlorophyll, system temperature, euphotic depth, KD490, which is its attenuation, far, light, and mount as input features, and we want to find primary production. And we get a decent fit. It's like kind of the same, not amazing. It's like, this is where I would have given up if I was not having a boss screaming at me, getting good results. But so we kept working on it, and we decided to also use depth as a parameter. In doing so, we actually got a pretty good fit. Now we have like 0.6, which is where it starts to get interesting. So we can actually predict, and this, once again, is in this upwelling region. There's a very hard area to work in. There are many caveats, many reasons, but yes.
By depth, you mean total ocean depth?
No, the depth of the observation.
Oh.
So we had these incubations down to 100 meters, so we just added that to the. So basically we're going with that, but that's a good question. So basically we're getting a much better fit here, which is great. But and then we can see here that depth is the feature that is has the most explanatory capacity, and then PAR, chlorophyll equally, and then down to almost nothing. But this shows that these models work well. We've done a bunch of testing, everything works well. If anyone is interested, we can have another conversation. That's another part. But going back to the question about depth, what's really cool with this now is that all of these parameters are defined at the surface, except the depth parameter. So basically, we have a time, so we have a location, and we have a time, and from that location and time, we get chlorophyll, surface, system temperature, blah, 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 except that. That means that we can put in any depth we want. In doing so, we can draw a gradient line. So now we can actually start to create depth profiles of primary production, which is this line. And as you can see, compared to observation, so this line is based on all the observed data. not even those data points that we're showing there. But we are actually able to recreate the profile, the depth profile of primary production in a really good way. Sometimes it actually has more scale. Sometimes it looks really good, and sometimes it doesn't look good at all. But we are able to do this, and this means that we now have a way of getting to primary production. This one was one of the worst ones. So it doesn't work all the time, but it works sometimes. And by doing so, we can now take all the observations of primary production we have in the ocean, and we include all the ones we have from this project, and we create a global primary production model. So this is depth integrated by creating these profiles in every grid cell everywhere. And this is a difference, shows a little bit of a different story than what these profiles or these maps normally do. For example, the North Atlantic bloom is much weaker in our results than they are in the traditional results. But then when I take the numbers we get, and I compare it to all the cruises in the North Atlantic bloom during the North Atlantic blooms, our data is much closer. Our results are much closer than what normally satellite data shows. And now if we zoom into one of these uploading regions, we can see that instead of having like a large plume of productivity that you normally or traditionally see when you talk about upwelling regions of primary production, so that most of the primary production happening very, very close to the coast. So you have this much more intense, narrow region. All right. The third thing I'm going to talk about is dominating timescales of variability. This is something we were working a lot. Basically, the problem is that if we are looking at chlorophylls from satellite data, it's very sparse. This is a typical time series from somewhere outside the coast here, and you can kind of see that it's going up and down, but it looks fine. But if I would just identify every day where we don't have data, it looks like this. So we have a 20% coverage in this case. It's very hard to work with that kind of time series. And the problem is that when we look at a time series, we can pick up different things. We can look at like a mean value, we can look at the long-term trend, and we can look at the variability. And our eyes are really good, or our brains are really good at picking up patterns. And we see this pattern about the seasonality really well. And now we are thinking this is a seasonal pattern. But is that the important timescales of variability? And that's what we wanted to figure out. And the question is like, how can we do that? How can we quantify this dominant timescale of variability in a very sparse data set? Because all the statistical methods that are used to do this don't like NANS. They don't like when there's no data. You can't use a Fourier transformation or EOFs or blah, blah. They're like every time I present this work, there's some statistician that have a pet method that we should use instead. And then we have a 10-minute discussion and they realize, yeah, I know, you can't do that. Which is as it should be, but I'm just saying this is the challenge. So what we've done instead is developing something we call the moving standard deviation saturation or MOS method. And I used to call it the cumulative sum method, and anyone who has a nasty mind can see that if you try to do any kind of abbreviation of cum and sum, it gets really, really bad. So I had to go back to MOS. But that was a much better way to do it. And the idea here is that you basically have a very small window, and you calculate the standard deviation in this window, and then you move this window across. It's basically a moving standard deviation calculation. And when we have a very small window, we see very little of the variability. And then when we increase the variability, the window size, the amount of the total variation we see increases. So in this case, we get a value here. This is like the average standard deviation of this window that's been moving. This one, we get a little higher, and then it gets higher, and then it gets higher. So basically, we can pick up where so this way we can create a curve that indicates something about the variability in this time series. And then we can go all the way out when it saturates. And in this area here, we're picking up all of the variability in this time series. So basically, a small window can see a little bit of the variability, a large window can see a lot of it. And then we just take the mean value and we plot that as a curve. And the nice thing with this is that when we have a slow, when we have the variability is on long time scales, the curve is the slope is kind of shallow, but then the higher the more frequent the variability is, the steeper the slope is. So the steepness of the slope explains what the dominating timescale of variability is. The other thing that's nice is that sparsity in the data doesn't matter. So the curve doesn't change if we remove up to 80–90% of the data. You still get the same curve. And we've done tons of testing around this, and there's tons of things that it can survive, which is great. So the slope varies with timescale, but it's robust for sparseness. And then what we can do is that we can take this value, basically what we've done is that we take the half saturation of the curve, and then we compare that. We find a relationship between that and the time scale. So we kind of scale it to a time scale from half saturation to time scale. And the nice thing with this scaling is that it doesn't matter how this figure looks like. We can use or the time scale looks like. We can use a sinusoidal, triangle, cutoff, sinusoidal, whatever. It all works. Doing so, we can start to calculate these curves. So in this case, we have chlorophyll and sea surface temperature, chlorophyll and sea surface temperature from different parts of the ocean. And depending on how the time series look like, the curve looks very different, and the slope of the curves becomes very different. And we can do this in different places. So the more of the seasonality we have, the shallower the slope gets, the more of just noise we get, the steeper it gets. And then we can do we've done this along a meridian, so we can see how it goes from high latitudes to low latitude to high latitudes. And that's what I want to show, that there's like a smooth transition in between different parts. There's no jumpiness or anything like that. It looks very, very nice. And then finally, we can do this with the entire globe. So this is the dominating timescale of variability for sea surface temperature and for chlorophyll. The first thing you can see is that they're very different. The sea surface temperature has much longer timescales than the biology. There seems to be a disconnect between the physics that are driving the sea surface temperature and the processes that are driving the biology. The other thing you can see is that in the coastal regions, we have very short timescales of variability. So there's a wealth of information to come out from these figures. And we can do a little bit of a blow up. So in this case, we can see the coast of the Gulf of Mexico and the coast of the US, of the east coast of the US and the Caribbean. And you can see that there is tons of small scale variability. Once again, every grid cell here is calculated individually, and they're based on 25 years of data. So if these structures were not real, they would disappear in noise. But like from 1 to 1 here, there's no like one grid cell doesn't know about the other. But now we can actually start to think a lot about what this means about the kind of processes that are working and so on. And of course, as we're here, we want to kind of continue. We talked to Gordon yesterday about the problems with the tide in this region. That is probably what you see up here. But that what's going on here, I have no idea. But now you can also see like how far out do you have a coastally dominating region? And what is the difference between this part of the coast versus this part of the coast versus this part of the coast? And if you're a phytoplankton living here, you might have a very different life than if you're living out here. And so there's a lot of questions, a lot of issues that we are just starting to work up on. And I think there's a lot of interesting things to work on when it comes to biogeography with this method. And then finally, going back to what we've been talking about, about upwelling regions. So the South American, the Hubble current in South America is one of the most important upwelling regions in the world. And if you use this method, you once again see this actually intense cells of high variability that we are interpreting as upwelling cells. So instead of having this like large scale, big blobby upwelling signal that's coming from high biomass, we do think that we're able to pick up these very intense and narrow regions where this is happening. So rather, because we're going from biomass to the actual processes. So what this means is that we have three different methods to estimate primary production or some kind of proxy to primary production or variability. They're giving very similar results, these narrow, intense cell-like products in different regions and using different methods. And going back to the process that we're talking about, this kind of fits with the idea that the growth and the biomass is disconnected from each other, and that we have this kind of temporal lagging. So we are now like trying to kind of tie it together and trying to think about it. But we think this is a better way of at least understanding primary production and biological production in these regions. And the reason why this is important to me, and this is from the question, is that this is one of my pet peeves for a long time. And this, I call it cartoon oceanography. The problem is that the way we understand the ocean, the way we understand the world, is from these kind of figures. This is what we're teaching, this is what we're showing. Like the large scale currents and everything is good. I've been working a lot about connectivity. And if you show something, if people think about that and you talk about like connection from the Antarctic to the subtropical oceans, they're like, yeah, but how can you cross the ACC? How can you cross the Antarctic Circumpolar Current? That's a wall. Because everyone thinks that these are rivers and every front is a wall. But this is how the world looks. This is the 12-degree resolution. So this is still coarse. This is still a crude estimation. We can go down much higher scales. But you can see there are no walls here, there are no rivers. Well, there's one river, but that one is kind of unique. Otherwise, it's just very, very fractured and very chaotic. So don't automatically assume that the cartoon oceanography is the true image of the world. And that's why I think this is important. All right, I'm just going to brag a little bit or kind of promote a next step that I'm working on that I think is extremely exciting. It's going to be exciting as well. And that's a new satellite that we're going to launch. The idea is that right now we have a number of different satellites, especially in ocean color, that are launching. The last one was PACE, which is amazing. With these hyperspectral satellites, so very, very high resolution in the spectral slope. So as I said, a camera normally has 3 colors. Traditionally, so red, green, and blue. Traditionally, these satellites have had maybe 5 or 6 colors. PACE has hundreds of colors. But basically, as a greedy scientist, I want higher spatial resolution, I want higher temporal resolution, and I want higher spectral resolution. So we've been trying to find the balance between these 3, and because each of them are being represented or being met by some instrument or the other. But to find that balance, we are going to launch a satellite in full glimmer. And this is a project out of the University of Hampshire, together with NASA Goddard and a bunch of other people. And it's a cheap satellite. I'm on the science team, but I'm a good friend of the guy who's the lead PI, so I hear all the dirty things that happen in the background, but I'm more focused on the science. And the idea with this satellite glimmer is to have basically very good hyperspectral resolution, a good spatial resolution, so 300 meters at nadir, and a temporal resolution hourly over the Gulf of Mexico. So this is not going to be a global satellite, it's just going to stare at the US, especially the Gulf of Mexico, and a very high signal to noise ratio. So we're trying to create a Goldilocks satellite. And what we want to use it for is things like coastal processes, high frequency fluxes of sediments, organic matter, episodic and seasonal river discharge, HABs, shelf exchange, a lot of words that are interesting in this building as well. So we're going to go in and trying to make this a coastal satellite. This just shows these questions, but spectral resolution. These are the spectral resolution the current satellites have, and this is the spectral resolution we will have. We're going to have a lot of data. And that's great, because that means that we can, in a much higher detail, pick up, for example, what kind of bugs we're looking at. This is an example of a HAB organism. I don't even remember which one it is. But you can see that from years, from a traditional satellite, we get these data points. And then one is a HAB. So the black here is a non-HAB species, and the red is the HAB species. And we're going to have to try to distinguish these two communities from each other with these 5 data points is very hard. If we instead do it via hyperspectral data, we at least have a chance. We at least can start to see the structure of the curve. And this is what we're going to stare. So 6 times a day at least when we got from Mexico. We're going to do a couple of days extra in the East Coast. So we're going to see the Long Island Sound a couple of days extra, which means that we're going to be able to see, combine this with, for example, PACE, and you will be able to get morning and evening images, which again can be quite interesting. And I'm going to stare a little bit at the Gulf of Mexico, the Amazon River blue, and then we have a couple of areas where we just want to do a deep water, blue water imaging, mainly for calibration. But it's also interesting to see how those are, partly because of the time budget that we have. And this is a scientist satellite. So we are able to control where we're staring. So if we have extra time and there is a big cruise or a big event, there will be possibilities to actually ask for time from the satellite and stare there, as long as we can see it in the disk. And these are the kind of products that we're looking at. These are the typical ones. We're going to try to get, these are the rate products that we're talking about today. And then we're going to have more of the very specific applications products. All right, thank you so much. This is basically where I was trying to go. I don't know how I am with time. No, it doesn't look too bad. These are so the people on the sides of the people who are working on for this big Primus upwelling region project, and then the other people are the ones that have been doing the other things. And that's my email if you're in any way interested in getting in touch, talking more. Thank you.
I think the first 2 questions go to Allison and Emma. Before.
Hi, thank you for the talk. I had a question about you were talking about using satellite and model images to sort of extrapolate where chlorophyll will be in the future, like kind of forecasting the future ocean. And I was wondering, in the case of harmful algal blooms, like what specifically are you looking for within those satellite images or models that would sort of like tip one off into, an area is more at risk or like an imminent force?
So very good question, first of all. And this is something I wasn't very clear about. What I'm talking about here is weather forecasts. Where you know that you might there are some meteorologists that will knock you over the head if you tell them, but we all assume that the weather forecast doesn't work, but they actually work pretty well. Like I know. I did my PhD in the meteorology department. I've had these conversations, and but in principle, the idea with the weather forecast is that we want to be able to say what's going to happen in 1, 2, 3, 5, 10 days. And that is incredibly useful because that means that we know it's not going to be true, but we know that we have a better understanding. Like if it says it's going to be shiny, you still bring an umbrella. But if it says it's going to rain, you're probably not going to have that outing and picnic. This is the same idea. Like if we know where the water masses are moving a couple of days ahead, and we see that there are conditions today that we know statistically will lead to a harmful algal bloom, or if we can see that we had Ravinia or some harmful algal bloom over there, if we know where that blob of phytoplankton will be in 3 days or 5 days, that means that we can talk to the scallop fishermen or the people, the aquaculture for oysters, and not to harvest at that point. Wait a couple of weeks. So we can use this as a warning system. We can also use it as a warning system for other pollutions, like at some points you might get some river effluents coming out with very high E. coli. we might want to close a beach and so on. So the idea is to actually being able to take what we see today and see how that will change, how that will evolve in a couple of days, and where it will go. And we think that has could be a powerful tool in that sense. Yes.
So much for taking the time to talk with us today. I was wondering more about the Lagrangian frame. You talked a lot about the particles, but
Yep.
what did you mean by that? Forgive me, I'm not physical.
Very good question. No, that's a very good question. This is where I was like trying to cut pieces. So basically, the two ways of the easiest way is to think about it as a float. Think about it as taking a rubber duck and throwing it in the water and then see where that rubber duck is going. That is how we do it in the ocean, right? Like we have a bunch of those experiments. So there's actually something called GDP, which stands for the Global Drifter Program, which is about 2,500 rubber ducky things that are moving around everywhere. That's very physical. That's 2,500 in the global ocean, so that's not much. So what I'm using instead is basically a way where I can take the output from an ocean circulation model. So these ocean circulation models are, once again, the same thing as the weather forecast models in the sense that they are simulating how the water will move. So you get what's called the velocity field. So basically it's just movement in X and Y and Z sometimes, but you see, you get these fields of how things are moving. And then you download all that data and then you just define a position. And you say, that's where my rubber duck is. And then using the information about how the water is moving, we follow that rubber duck in the ocean model. And we can see where we can get a track of how it's going. And that track is what we call the trajectory. So basically we are able to kind of predict or follow a water mass and seeing how that works. So now we can think of the changes in the ocean from the perspective of sitting on the rubber duck. Now it has to be a big rubber duck and seeing what's going on around you. That's called the Lagrangian frame. The opposite to that is the Eulerian frame, which is just sitting stuck somewhere and have the water moving, slushing back and forth under you. And that is normally how we think, how we do exercise. Like basically we go out the same point once a week and we measure the same thing, week after week after week. But the problem is that the water we measured last week is now halfway to Nova Scotia. So we're measuring new water. So that there is a lot of benefits in the Eulerian frame as well, but there are also disadvantages. Great. Other questions? Yes.
You might have said this, I'm not sure. Are you able to identify exactly which species is causing the HAV based on their reflectance to your satellite?
Ish. There are some organisms that can be detected. There are some algorithms that can do this. And the most important, the one most famous is Carina brevina. I think it is. I can't deal with the Latin names. I'm not enough of a biologist for that. But basically red tide, because that has a very distinct color. Unfortunately, the ones that are most dangerous are the ones that we can't pick up optically. We can't see it from the color. So we had a project. We have a kind of zombie project at PML. It's a zombie project. I heard some of you were writing, had a proposal writing course. I'm going to give you one advice when it comes to proposal writing. And this is a very, very important advice. When you write your proposal, submit it in time. Because we wrote this, we spent months writing this proposal. It was awesome. We thought we're going to use machine learning to pick up exactly those organisms you was talking about that we can't normally see. We had all these great ideas. Everyone was very excited. And the guy who was supposed to submitted it 10 minutes too late. No, 5 minutes too late. 5 minutes. And yeah, that was not fun. So don't be out last minute. There are so many things that can happen. Yeah, anyway, so yes, we can, we have ideas, but there are definitely things that we need to think about there. And the other thing I think is very important that we've been starting to think about is stuff like E. coli or effluents from rivers, because in Europe, that is a big issue, but it's going to be, and it's a big issue here too. It's like people get sick because suddenly you have water coming from a water treatment plant or a water plant that's not treating the water and so on. So then there's a lot of questions around that. Yes.
What about tracking trichodesmium?
Good question. We are, as soon as we can get a signal, that would be fine. I think we're suggesting that we can do it. I say that, but would be fantastic to do actually. Yes.
A practical question. You made a point that satellite data and Lagrangian approach gives more and more focus that there are highly localized productivity spots very close to shore.
Yep.
Well, we have a lot of boats that we deploy locally and around the world in inshore waters. Are they a waste of time? That's a real practical question. They collect data on, for example, the Long Island Sound Project collects every couple of weeks data with depth, chlorophyll and everything else to see whether isothermal conditions are correlate with the onset of phytoplankton boom. Is that information still important?
Yes, absolutely. So what you can get from those from the boats is really mechanistic relationships and these connections. Like whatever data there is, we need the data definitely. When it comes to observations, I think that we need to think more about how to do this. One thing I'm promoting right now when it comes to these coastal processes is that what the satellite data, the satellite approach doesn't do well is specificity, is regionality. We have these algorithms that are global for the entire ocean. The entire world that works really well, or they work okay for chlorophyll. They don't work at all for CDOT. They don't work well for turbidity. So for doing that, we need to have much more regional algorithms and regional data, and that is where we need these field campaigns. So I think it's going to be the coastal ships, the coastal cruises, etc. It's going to be even more important for that. What I would love to have on that ship or those boats is a simple radiometer, because if you can get a radiometer on those boats, and then I know Maria Chocho at City College New York had done that, and there are people up at Cornell that's doing it too. That means that we can get data when you go out, and you can get the optical data from the field, and then we can use that to the algorithm. So we're kind of helping each other that way. But those two things will always be in unison. I'm actually more. I think it's more important to have the boats than the floats, to be honest. Yes.
When using passive remote sensing, like ocean color, for instance, do you account for biases in like seasonal light distribution, sea ice cover, and like average areas where cloud cover maybe more be top versus on?
Yes, we do. Poorly or good, depending on how close we are. But yes, we are. So basically, when we are we get what we call the top of the atmosphere radiances, and then which is basically what we measure with the satellites. And then we use algorithms to go from those to what we think is at the surface of the ocean, and that is called atmospheric correction. There are very, very different approaches to do that. So if you are interested in doing passive remote sensing, you should definitely go in and see how these things work. Because the Europeans are using different atmospheric corrections than what the Americans are using. And there are a number of different European groups that are doing it with different foci. And especially for statistics, especially for, as you say, like ice cover and so on, this can become like a first order issue. So understanding what it has been masked out by the atmospheric correction algorithms, because they are the ones who do the masking out of ice, of clouds, of glint, which is basically the sun is shining right up into the instrument, etc., etc. It takes a little bit of time to figure out, but then once you do that, you can even try to process a couple of fields yourself to see how that works, because everything is online.
Amazima.
Yes.
Two questions and a comment. In your MOS approach, how different is it from the EMD, the individual molecule decomposition? And when you tried it, since you did not introduce noise, how sensitive is it to noise? And my comment would be those hotspots, especially around the Hamburg current, look like they were controlled by tropical, the coastal tract waves.
Yep. Good questions. To the first one, EMD, I need to go back and think about it. Everything looks very different. We've kind of gone in spirals there. The second one, when it comes to noise, we see noise as signal. So we're not removing any noise. Basically, if you have a white noise signal like this, you're just going to get a very, very short timescale variability. But that is like kind of part of the theory of what noise is. We are not, because we're assuming that the noise is still signal in the sense that that is what the satellite sees. We could put a low pass or high pass filter in different ways. We've been playing around with that, and that gives different results. But in some ways, what we're seeing here is that we're looking at what time we saturate the data. So low pass filters doesn't make too much of a difference. High pass filters, so basically cut at a shorter time scales, would probably let us see things where we would be able to see structures that are now disappearing because the seasonal cycle is swamping the data. That is something we've been thinking about. We haven't gotten to do yet, but I especially with sea surface temperature, that would be very interesting. And your comment is absolutely right. I mean, you can see that in the Caribbean, you have these very narrow bands of blue around the islands. That is just the coastally trapped waves and other kind of advective processes that are happening. So when I show these three different ways of looking at the data.
Sorry, yeah,
I know where. So we have the Lagrangian approach, we have the dominating timescale approach, and we have the random forest. The reason why we can't just compare them directly, and the reason I was cheating by using different regions when I showed it, is because they're showing different things. I think that the Lagrangian primary production system is the most like really are estimating primary production in a very correct way. The random forest regression method is picking up signals that are much less. It doesn't know about advection. It's still kind of based on biomass, but it's not as precise. And then this timescale variability is absolutely not picking up advection. It just looks at it from a Eulerian perspective. On the other hand, and then the timescale variability is a 25-year data set. The primary production data, the random forest regression approach is a 1-month data set, and the Lagrangian approach is a 1-year data set. So everything is slightly different, but we're still kind of picking up these intense growth cells that I think very interesting.
I think we had one last question. Did you want to ask a question?
Yeah, but so when you mentioned that satellite one flush up, and I was under the kind of impression that the sort of like, I don't remember what it's called, the zone where satellites orbit is like becoming increasingly crowded.
Yeah.
So I was wondering if you're going to a point where you have multiple satellites accomplishing similar but different but like related similar goals, how you're prioritizing which ones to keep up there, if there's like a need to take previous ones down, and how that sort of fitting.
So the 3 levels.
Sorry.
Yeah, there are 3 levels of where you can launch satellites. You have the lower orbits, which are where you have the Starlink satellites, these big constellations. They will drop in a couple of years because they're almost touching the atmosphere. So they're cheap and they're easy to launch, but they're also very fungible. And then you have the next level, which are the polar orbiters, the real polar. And the first part, that's where we have a lot of crowdiness, and the astronomers are very sad and angry, and so on and so on. The polar orbiters are the Paces and the MODIS and Sea-Waves and so on. They're higher up, and they're moving like this, so they are much more. They have different orbits, but they are they basically when the Earth is spinning like this, they are spinning around the Earth. So they are spinning around the Earth like an hour. Each orbit is about an hour. And then because of that, you can get like images everywhere on a daily or 2-day or 3-day basis. These are the normal satellites we're using in science. What we are going to launch, or we hope to launch, well, we have the money, we just need a a ride, is what's called a geostationary satellite. Is we're going to stare at the same place all the time, which is what the weather and the communication satellites does. That is much, much, much higher up. There's like 30,000 meters up. So they're very expensive to launch because they're all hanging over the equator and they are spinning at the same time orbit as the Earth. That is why they're seeing the same spot all the time. And that is not at this point very crowded because it's just very expensive to launch. So we are going to piggyback on a communication satellites or something like that. We haven't gotten our ride yet. NASA is trying to negotiate that, but there is that's the idea there. But yeah.
Well, you're gonna have to sell it, you're gonna have to say you're staring at the Gulf of America.
I know
yeah.
that is exactly it.
All right, on that note. Thank you.
Thank you for listening.